Let's look at our schedule, see what we're supposed to be doing. So this is February 3rd. We're supposed to finish chapter 2 and start chapter 3 and do some lab stuff. I got some lab stuff set up over here. That stuff's not for us, that dissecting stuff. That's a different class. Then I got his name's Carl, by the way. We've got that thing going, so we can play with that later. I know how to turn it on and tiny bit more than that, but we can try to figure it out some later tonight if we have time. What else? So every Monday night there's Moodle stuff due, right? Moodle quiz and short answer questions. And so, I don't remember if I said it to the whole class, but on those short answer questions, if you answer one and you think the answer is right, but Moodle said it's wrong, that's just because Moodle can't think. All it can do is compare your answer to the answer stored in Moodle. And in some cases, I've got several versions of the answer stored in there. But students can still come up with another way to answer it that's not wrong, but it doesn't match any of the answers in there. It has to match exactly, letter for letter. If you put an S on the end of a word to make it plural, Moodle will say that nope, this doesn't match, so it's wrong. So after the deadline, I go in and look at all the answers that Moodle counted wrong, and I'll check them and make sure. In many cases, they're fine, so I'll change the grade. And then, there's one question about which kind of section would something, I don't know, but the answer was supposed to be frontal plane, and more than half of the class put the word coronal plane, and that's not incorrect, but I have never said that word. When we covered that chapter, I used the word frontal. The word frontal is what we used on the PowerPoint. That word coronal is not even on the PowerPoint anywhere. So I don't know if you Googled that to get that answer, or that word is probably in the textbook. So that's fine, but if you do that, I'd rather you don't Google those questions, because you're more likely to get an answer that is not the answer that I expect, even though you might get a correct answer or an answer that's not wrong. And also, if I would put that same exact question on an exam in the multiple choice, coronal is not going to be one of the choices. So you better know that frontal means the same as coronal. So that's another reason. But so all of you put coronal. I changed that to give you the points for it. That's fine. But I think frontal is used more than the word frontal is used more than the word coronal. I've never heard anybody use the word coronal in any anatomy classes I ever took, or any other instructors. Okay, well, we need to continue on chapter. Wait a minute. Let's look at the schedule again. Yeah, we have an exam scheduled next week, right? Chapters 1 and 2. So that will be multiple choice, probably 50 questions. Most of my exams are 50 questions. Multiple choice and matching, very similar to the sample questions. Some of them might be exactly the same as the same sample questions. So I think if you can answer all those sample questions and you know and you understand the sample questions, don't just memorize those questions and their answers. That's not the way to use them. You need to understand why the answers are what they are. But if you can do that, then you should be ready to do just fine on the exam. And there is a study guide. I forgot it was on there until one of you emailed me about it. There's a study guide on Moodle here somewhere for that exam. Where are you? Yeah, there, lecture exam number 1 study guide, chapters 1 and 2. So, so that's under last week 3. This is week 4 already. So that's just a list of words and concepts that you should know. So that might be helpful. Just look it over. If there's anything on there that you have no idea what it means, you better look it up because it could show up on the exam. But I think everything on that study guide I took from the sample questions when I made this up several years ago. I think I made this in 2018. Because some students asked me for a study guide. Okay, so let's go back to chapter 2, finish this up. So this is a chapter on chemistry. So we were talking about atoms and elements and protons and neutrons and electrons and that octet rule. So the electrons are arranged 2 in the first electron shell, or only 2, up to 2 in the first shell, 8 in the second shell, 8 in the third shell. And atoms like to have their outermost shell full. And then the atom is what we call stable. It's happy the way it is. Otherwise, it's reactive and it will undergo chemical reactions to hook up with other atoms to form molecules. And as they do that, they're getting their outermost electron shell full. Now I'm teaching 4 classes this semester and teaching chemistry in all 4 of them, and I get so confused. I can't remember where I left off in this chapter. Did we get this far? Did we talk about acids and bases? No?
You touched on it. You were skipping around a lot.
Yeah, I remember. I'm sorry. I felt bad the way I did. I was skipping around too much. It's probably confusing. But so I talked about water and hydrogen bonds and how why insects can walk on water, surface tension. Did we talk about that?
Mm-hmm.
And then solutions, and we talk about any of this, chemical reactions. I don't know, probably quit around here somewhere then, right? Okay, well, chemical reaction is just a process in which um, two atoms or molecules come together to make a bigger molecule, or else molecules are broken apart. Um, and the but this word metabolism, very important word in physiology. It just means all the chemical reactions going on inside of an organism, like your body. Lots and lots of chemical reactions going on all the time. Your digestive system is breaking molecules apart, and the cells of your body are making new molecules all the time. This law of conservation of matter, you don't need to know that. It just means matter cannot be created or destroyed, but it can be rearranged. And so uh, um, these are examples of chemical reactions, the way they're written. Um, the, the way a chemical reaction is written, these are examples. And on the left hand there, there's the before the reaction is the reagents, or better word is reactants. The word reactants is used more. And then after the chemical reaction is the products. So this could be, for example, two molecules. This one is methane, CH4, and this one is just O2, oxygen. So those are the reactants. And then the arrow represents the chemical reaction. And then after the chemical reaction, in this case, there's carbon dioxide and water. So that law of conservation of matter means that the elements on the left side of the arrow, the elements in the reactants, will always be the same as the elements in the products. So new elements, or elements don't disappear, or elements don't change into other elements in a chemical reaction. They're just rearranged. So in here there's carbon, hydrogen, and oxygen on the left, and carbon, hydrogen, and oxygen on the right. They're just rearranged. Same here, sodium plus chlorine. They come together to make this molecule, sodium chloride, which is salt. But there's the same elements on both sides. This is photosynthesis, what plants do. They use light energy along with carbon dioxide from the air and water from the soil, and they make this bigger molecule, glucose, and they exhale oxygen. Like we exhale carbon dioxide, they produce oxygen as a byproduct. But again, only carbon, oxygen, and hydrogen on the left, carbon, hydrogen, oxygen on the right. There would never be nitrogen would show up here if it wasn't here. There's always the same ones on the left as on the right. Okay, energy, the word kinetic, kinetic energy is just energy of motion. So anything that's moving, we say it has kinetic energy. If I were to drop this to the floor, it would fall, and as it moves, it would have kinetic energy. Even the atoms and molecules, I think I said last time, the atoms and molecules in a liquid or in air, they're always moving, bouncing off of each other. That's a kind of kinetic energy. Potential energy is energy that's not doing anything right now, but it could cause something to happen. It could cause something to move. That's what the word potential means. It means the ability for something to happen. So an example of potential energy is, you'd say there's potential energy in this remote sitting there, just because if I would let it fall, it would accelerate downward. So just the fact that it's up off the floor, 3 feet, gives it potential energy. So there's the potential that it could move. But there's many kinds of potential energy, including chemical energy that is stored in the bonds holding atoms together in molecules. It's a type of potential energy. And so, in many types of chemical reactions, as the molecule is broken apart, some energy is released um, from that molecule. Energy that was stored in those bonds. Law of conservation of energy. Energy also cannot be created or destroyed, but it can change from one form to another. Like electricity can be converted into light. That's what a light bulb does. It converts electric energy to light energy. An electric motor converts electric energy into kinetic energy. It makes the motor spin. If I bend my elbow, that's using the chemical energy in by converting the chemical energy from the food I eat into the energy, or converting the chemical energy in the food that I eat into kinetic energy, energy of motion. So exergonic and endergonic reaction. X means out, end means in. This just refers to energy either being released during a chemical reaction or energy being used up in a chemical reaction. So an example of an exergonic reaction is just burning something. Burning is just a chemical reaction between some kind of fuel and oxygen. But that chemical reaction releases heat energy. So we say it's exergonic. When nutrient molecules like sugars are broken apart in our cells, that releases energy, and that's the energy that keeps your cells alive. It's the energy that the cells use for everything. You guys ever do anything with plaster of Paris? Mm-hmm. It's a powder, and you mix it with water, and then it hardens, right? But what else happens? That that heats up as it hardens. That's not just the water evaporating out of the plaster of Paris. It's a chemical reaction between the water and the plaster that causes it to harden into a solid. But it's an exergonic reaction, so it releases heat. If you touch that, you feel it heat up as it hardens. Endergonic reaction is just the opposite. Some chemical reactions absorb energy as they happen, or they won't happen unless you heat it up. That's why in chemistry labs they got these Bunsen burners and hot plates and things. To get some kind of chemical reaction to happen, you have to heat it because they're endergonic. The chemical reaction uses energy. And that energy will become stored in the bonds in the molecules that are formed in those chemical reactions. This activation energy required to start a chemical reaction. I think I talked about that last week when I said something like, for example, if these two are not that. This said that hydrogen is flammable. I remember talking about the big airships and the one that caught fire, and we talked about that, right? Yeah. The hydrogen gas is very flammable, and oxygen, O2, and like I said, burning is just a chemical reaction between something like hydrogen and oxygen, and it releases heat. But I also said that these are both stable because when these two hydrogens are together, they're sharing electrons, and now they got their outer electron shell full, so they're stable. Same thing with O2. The reason those two oxygens came together to form a molecule with covalent bonding is to get their outer shells of electrons full, so now they're both stable. Stable means they're not reactive. So how could burning happen between those? Well, it's because if you just mix those two together, they won't burst into flames or anything because they're stable. But if you put a flame to that, put some fire to that, it'll heat up the H2 and O2 molecules so that they'll separate. These will come apart into two H plus or two hydrogen atoms, and this O2 will come apart into two oxygen atoms. Now these are unstable, and now that when they come together, there'll be a chemical reaction and release more heat because it's burning, and that heat will cause more H2 and O2 to come apart. So it's like a chain reaction. All it needs is some activation energy to get it started, and then it'll keep producing more heat to make more of them come apart and burn. Okay. Enzymes. Where are we? We didn't talk about. Yeah, we didn't talk about proteins and carbohydrates yet, did we? No. So enzymes. Enzymes are special kinds of protein molecules. We'll talk about what exactly a protein molecule is in a few minutes. But enzymes are molecules, protein molecules that cause chemical reactions to happen in living organisms like your body. There are many important chemical reactions in your body that would not happen if there were not enzymes causing them to happen. And for example, digesting food. Enzymes are the molecules that like break the large food molecules down into smaller ones that can be absorbed and used in your cells. So we have lots of different kinds of enzymes. Each enzyme does only one kind of chemical reaction, and the enzyme is not used up in the chemical reaction. It's not one of the reactants. It just causes. It'll just like an enzyme will take two molecules, stick them together into one molecule, and then release them. And it'll grab two more, put them together, release them. And the way they do it is it's described here. It's kind of a weird diagram, but it lowers the amount of activation energy required to get the chemical reaction going, and it's compared to they're comparing the activation energy to get the chemical reaction started to the amount of energy it takes to push a rock up this hill. Once you get it to the top, then it'll continue on down by itself. Kind of like, the energy you have to put some energy to the hydrogen and oxygen to get it started, but once it gets started, it takes off on its own. But with an enzyme, it takes less energy, so the hill is not as high. It doesn't take as much to push that rock to the top of the hill to get it going, to get the chemical reaction going. We'll talk more about enzymes in a minute. Okay, chemical reactions, synthesis means to make something, so that would be a chemical reaction that makes molecules. Decomposition, that's a chemical reaction that takes a molecule apart. Exchange, well, there's examples here. Yeah. Synthesis, putting two things together to make bigger molecule. Decomposition, taking a molecule apart. Exchange is just rearranging. So on this molecule, the A and B were together, and then the C and D were together, but after the chemical reaction, they're just rearranged. Now the A is with the D, C is with the D. The letters A and B are not symbols for elements, they're just for examples. Reversible has arrows going both ways. So that's a reaction that can go either way depending on the circumstances. And then this one is redox reaction. Don't worry, you don't have to know this, but this is really important in biochemistry. An oxidation reaction is one in which a molecule is losing electrons or losing hydrogen atoms, either one. A hydrogen atom is just one proton and one electron, so if a molecule loses a hydrogen atom, it's losing an electron, but a proton along with it. So that's called oxidation. Reduction is the opposite, a molecule gaining electrons or hydrogen atoms. And it seems backwards, doesn't it? It seems like reduction would be losing something, but in chemistry, reduction means gaining electrons and hydrogen atoms. So a redox reaction is a reduction reaction and an oxidation reaction together, so that the atoms, the electrons or hydrogen atoms that are lost by one molecule are gained by the other. So it's just transferring electrons or hydrogen atoms from one molecule to another. So that's a way of it's transferring energy from one molecule to another by shifting electrons from one to the other. But we're not going to worry about that in this class. Okay, well, dehydration, synthesis, and hydrolysis, decomposition. In a lot of kinds of molecules, like proteins and starches and long molecules that are made up of a bunch of small molecules linked together. When two small molecules are linked together, and they're called monomers, and they'll come together to make a polymer. Polymer means many molecules together. Like if these were two sugar molecules, and they both have this oxygen and hydrogen attached to them, and when they come together to form a bond like this, now there's only an oxygen where there was an oxygen and hydrogen here, oxygen and hydrogen here. What happened to the other oxygen and the two hydrogens? They become a water molecule. So when these two molecules are bonded together, they form a water molecule. So that's called dehydration synthesis. Dehydration means losing water. So a water molecule goes away when that bond is formed. The opposite of that is hydrolysis, decomposition. To break the bond between these two molecules, there has to be a water molecule present that can break apart, and because when this bond is broken, there has to be a hydrogen added to that oxygen, and an oxygen and hydrogen added to the other molecule. So hydrolysis means breaking water molecule apart. So this is called hydrolysis, decomposition. Okay, organic This word organic chemistry. If you major in biology at a university, you have to take a whole year of organic chemistry. And the organic chemistry is the chemistry of carbon compounds. The word organic has different meanings. If you go to Walmart to buy some bananas, you got choice. There's bananas, and then there's organic bananas. And what's the difference? Organic ones cost more, right? But the organic in that case means natural. So the organic ones are grown without chemical pesticides or artificial fertilizers and stuff. Organic can sometimes mean lifelike, but in chemistry, organic means carbon compounds. And in the early days of chemistry, the chemists kept finding these certain kinds of molecules that seemed to only exist in living organisms, proteins. They never saw proteins in a rock, or they were only in living organisms. And so they called them organic. But in order to be considered an organic compound, it has to have at least one carbon atom and at least one hydrogen atom attached to the carbon. It should be on there somewhere, but it's not. The carbon dioxide has carbon, but it doesn't have hydrogen, so that is not organic. All these are inorganic. They don't have carbon and hydrogen in them. So they're all called inorganic. There are many kinds of organic compounds, and that's partly because carbon is an atom of carbon. It has the nucleus, and then it has 6 electrons, 2 in the first shell, and then 4 in the second shell. That second shell would like to have how many? 8. It can hold 8. It would like to have 8. Atoms like to have their outermost shell full, so it needs 4 more electrons to make it stable and happy. And how can it do that? It could do it by sharing electrons with other atoms, like if it would. Here's a helium. It has only 1 electron, but if it shares that electron with the carbon, now it's as if the hydrogen has 2, and the carbon has 5 now, but it would have to do that with 4 hydrogens, sharing electrons with 4 hydrogens to get its outer shell full. So carbon can form 4 covalent bonds with 4 other atoms, and that they could all be hydrogen. They could be a hydrogen, an oxygen, a nitrogen, something else. And so there's many different kinds of organic compounds that can form. Okay, water. We talked about hydrogen bonds and the weak attraction between water molecules in liquid water because they are polar. And the positive end of one water molecule is attracted to the negative end of another. Those are called hydrogen bonds. And that's what causes surface tension. It's like a skin on the surface of water that insects can walk on that. But it also gives water some other important features, like water has a high heat capacity. It means water absorbs a lot of heat. It takes a lot of heat to raise the temperature of water compared to other liquids, because if you put water on a stove and there's heat going into that water, the heat is just the motion of the atoms or the motion of the water molecules. The faster they get going, the more heat they have. And so the heat from the stove is causing the water molecules to move faster, but at the same time, those hydrogen bonds are pulling the water molecules toward each other, so that's resisting their motion. So it takes more energy, it takes a lot of energy added to water to get it to heat up, compared to something that's nonpolar like gasoline or oil. If you'd put them both on a stove and a pot with the same amount of heat going into the water and going into the oil would heat, or the oil would heat faster than the water, because the oil doesn't have hydrogen bonds and the water does. Yeah, there's heat capacity. Heat of vaporization. It takes a lot of heat to evaporate water too, because if I have a container of water, all these water molecules in here, they all have hydrogen bonds attracting them to each other. And when water to evaporate, it means the water molecules are leaving the liquid and going up into the air and becoming water vapor. So it takes a lot of energy for a water molecule to evaporate, because it has hydrogen bonds holding it back. So a water molecule has to get going pretty fast to break loose of the liquid and go free into the air. It's kind of like an airplane trying to take off. There's gravity holding the airplane down. If it's trying to take off, it has to get going pretty fast to break free of or overcome the gravity and become airborne. And that's why you feel cold when you're wet, because every water molecule that evaporates from that water is taking heat energy with it. So the water that's left behind is cooler and makes you feel cold. An unusual feature of water is that it's when it freezes, it becomes lighter, less dense. So ice floats. Other kinds of substances that exist as both liquid and solid, the solid is heavier, but in water, the solid is lighter. That's because in liquid water, the hydrogen bonds are pulling the molecules together, making it denser, more and heavier. Okay, enough about water. Oxygen, we all need that, right? For the reason we inhale oxygen is because there are these chemical reactions in just about every cell of your body for breaking down nutrient molecules like sugars to get the energy out of them, and those chemical reactions require oxygen, and those chemical reactions are called cellular respiration. Those same chemical reactions produce carbon dioxide as a waste product, and we exhale that. But we always have some carbon dioxide in our blood. That is that helps regulate the pH of your blood, how acidic your blood is. If you don't have enough carbon dioxide in your blood, your blood will not be acidic enough. It'll be too basic. This word electrolyte is important in physiology and in medicine. Um, what that word really means in chemistry is a molecule that when you put it in water, it comes apart and forms ions. So table salt, any sodium chloride. When you put it in water, the sodium ions and the chlorine ions separate. So that, therefore, salt is an electrolyte. But in medicine, if the word electrolyte is usually used to refer to the ions themselves, not the sodium chloride, but the ions, the sodium or the potassium or the calcium in a patient. And that's very important, especially in little kids. The kids in a hospital, one of the things that's monitored is their electrolyte levels. They've got to have the right amount of sodium and potassium and calcium in their body fluids, in their blood. And because little kids are so small, if they have diarrhea or vomiting, they can lose a lot of electrolytes that way, and their electrolyte levels can change really quickly, and that can be really dangerous. Okay, a salt. So we call this stuff sodium chloride. We call it table salt, but the word salt is a more general term in chemistry that can mean a lot of there's a lot of different salts. There's sodium chloride, there's magnesium chloride, magnesium sulfate. Those are all considered to be salts. So the electrolytes made of oppositely charged ions. This part about being formed from an acid and a base, I'm not really sure what that even means. But these are all important ions here in human physiology. You've got to always have the right amount of them, not too much and not too little. Okay, if this is pure water, I didn't talk about this stuff. No? Okay. I did?
A little bit.
Okay. So I've been repeating, or probably I was jumping around, but I won't jump around anymore. So this is pure water, just liquid water. Water molecule, water molecules are H2O, right? When those water molecules are bouncing off of each other all the time, sometimes one of them breaks apart. And when that happens, it'll break apart into these two ions, H plus or hydrogen plus and OH minus. This is called hydroxide ion. So this happens all the time in water. Sometime later they might come back together, or this H plus might come together with this OH minus and form a water molecule again. So there's always water molecules breaking apart and then coming back together in liquid water. So at any given time, any given moment, in a container of water like this, there might be like 1,000 water molecules are broken apart. The rest of them are in one piece. But if there's 1,000 water molecules broken apart, then there will be 1,000 H plus and 1,000 OH minus, right? Because you get one of each when a water molecule breaks apart. So there'll be equal in numbers of H plus and OH minus in pure water. That means that because these are equal amounts, that means that this is not acidic or basic. It's not an acidic solution or a basic solution. So this word acidic just means that a solution has more H plus than OH minus in it. So if we would add H plus ions to this pure water, then it would become an acidic solution. And a compound that if we add it to water, it adds H plus, that's called an acid. That's what's meant by the word acid. So for example, hydrochloric acid, HCl. this this molecule is held together by ionic bonds, and it's an electrolyte. So that means if you put it in water, it comes apart, right? So it comes apart to form H plus ions and fluoride, fluorine ions. So it's adding H plus. If you put that hydrochloric acid in water, it's going to add H plus, make that solution more acidic. Nitric acid, same thing. HNO3, put it in water, it comes apart, and it adds more H plus. So it makes the solution more acidic. So it's an acid. The word basic in chemistry means the opposite. A solution has more H plus than H, or more OH minus than H plus, or less H plus than OH minus. And something that we put into a solution that adds OH minus, like sodium hydroxide here, put it in water, comes apart, adds OH minus to that solution. So this is a base. That's what Li is, Ly, sodium hydroxide. Magnesium hydroxide, milk of magnesia. Put it in water, comes apart, adds OH minus. So that's a base. So that makes the solution more basic. Adding OH minus to a solution is kind of the same as removing H plus, because if you add OH minus to this, they will attach to any H plus in there to form water molecules, so would make less H plus than OH minus. The word alkaline means just about the same thing as basic. So an alkaline solution is the same as a basic solution. A chemistry professor would say, no, it's not exactly the same. It's a little bit different, but for our purposes, alkaline and basic mean the same thing. Okay, so pH. You've all heard of pH. The pH is just a measure of how acidic or basic a solution is. So it's a measure of like how much H plus is in a solution, and it's the pH number can be from 1 to 14, and right in the middle of that range is 7. Halfway in between, and so we say, like I said earlier, pure water is it's neither acidic nor basic. It's in between. So it has we say it's pH neutral. It's not acidic or basic. And if a pH is less than 7, this seems kind of backwards, and it is. If the pH is less than 7, that means more H plus. And that's just because of the weird formula that they use to calculate pH. You don't need to know that formula, but turns out that low pH means high H. And these square brackets like this in chemistry, that's shorthand for the concentration of that stuff in a solution. So this means the concentration of H plus ions in a solution. So for example, like how many H plus per milliliter of that solution. So low pH means more H plus, which means acidic solution. High pH, greater than 7, means less H plus and basic solution. So that's summarized here in this bottom line. What you need to know. Low pH means high H plus, which means acidic. High pH means low H plus, which means basic. This is important in biology because the pH of a solution in which chemical reactions happen can greatly affect the chemical reactions, mostly because enzymes, there's protein molecules that cause chemical reactions to happen. Enzymes are very sensitive to pH. If they're in the wrong pH, a little bit too acidic or a little bit too basic, those enzymes will just stop working. So this is why it's important that pH of the body fluids of a person can't get too low or too high. Okay, here's some familiar solutions and their pHs. These are all. I'm not sure why you got the red and blue, but so here, right in the middle here is 7. That's pure water, distilled water. Equal amounts of H plus and OH minus, not acidic nor basic, but anything below that is acidic. So cow's milk, just a little bit below 7, so it's a little acidic. Cabbage, tomato juice. Tomato juice has citric acid in it, right? So it's acidic. Apple juice, and gastric juice. What does gastric mean? Stomach, right? So that's the liquid in our stomach that has hydrochloric acid in it, a pretty strong acid, pH 2. If you didn't have a thick layer of mucus on the lining of your stomach, that acid would eat holes through the tissue of your stomach. That's what a stomach ulcer is. Okay, these above 7 are basic. So egg white's a little basic. Sodium bicarbonate, this is baking soda, sodium bicarbonate. It's a base. Milk of magnesia, household ammonia, soap is a base. Bleach is a pretty strong base. My dad used to, when he had an upset stomach, he'd put baking soda in water and drink it. Because if his upset stomach was caused by the acid in his stomach, then that's a base. It would help to neutralize that acid, because if you mix an acidic solution with a basic solution, you get a pH somewhere in between. So if you add this base, baking soda, to stomach acid, it would raise the pH of the stomach liquid, so it's not so acidic. Same thing with milk of magnesia. That's why people take milk of magnesia for an upset stomach. Something all these bases have in common is they feel soapy and slimy if you get them on your hands. If you mix baking soda and water, it feels soapy. You ever get ammonia or bleach on your hands, it feels slimy and soapy. It's kind of hard to rinse it off. Acidic solutions don't feel like that. Okay, so buffers. A buffer, a chemical that combines with excess H plus or H minus to help minimize pH changes in body fluids. So that, in other words, if a solution has a good buffer in it, then if some acid or base is added to that solution, the pH won't change, because the buffer, it's like the buffer soaks up the extra H plus or OH minus. So we have several buffer systems in our bodies, and this is one of them, the bicarbonate and carbonic acid. You know, we always have this stuff in our blood, and so we see this chemical reaction arrow goes both ways. That's one of those reversible chemical reaction. It'll go either way depending on the circumstances. So if you, what do you about lemon juice? You think that's acidic or basic? We tested that last week, didn't we, with pH paper? And it's quite acidic, right? like pH of 3 or something. So what would happen if you drink 3 glasses of lemonade? The pH of your body fluids has to be, the pH of your blood is, the normal range is 7.35 to 7.45. It's a very narrow range. If it gets just a little bit out of that range, below or above, you're sick. You're either in alkalosis or acidosis, they say. So if you drink 3 glasses of lemonade, that's going to be adding a lot of H plus to your body fluids. Does that mean the pH of your blood is going to get lower and lower and lower because of that lemon juice? No, it's because you have buffers. You have good buffers in your body fluids, in your blood. So if you drink all that lemonade, adding a lot of H plus to your blood, you've always got a lot of this bicarbonate in there that will combine with those extra H plus and just make these carbonic acid molecules. So it's like soaking up the H plus, getting it out of the solution. If you don't have enough H plus, or you eat something that's very basic, and it won't raise the pH of your blood. It won't make your blood too basic because you always have a bunch of these carbonic acid molecules in your blood. So if you need more H plus, these will come apart and just add more H plus. So that's why this arrowhead both ways. It'll go to the right if you have too much H plus, but it'll go to the left if you need more H plus. Okay, or you need to talk more about organic chemistry. Okay, yeah, here's where it says that at least one carbon atom with at least one hydrogen atom bonded to it to be an organic compound. Otherwise, it's inorganic. Yeah, I already talked about this stuff. Okay, practice questions. Okay, there are a lot of types of organic compounds, but these listed here are large, important organic compounds that make up much of our bodies, and also other organisms. And these are also what make up much of the food that we eat, right? Because what do we eat? We eat other organisms that are made of the same kind of stuff as we are. So carbohydrates, lipids, proteins, nucleic acids. So we'll look at each of those a little bit. Lipids are fats and oils. Nucleic acids are DNA and RNA. But we'll look at each of those for a minute. This is important to understand what these things are. But this is generally organic compounds. Many of them have these long chains of carbon atoms, and with other stuff attached. This one has only hydrogens attached, and this one there's 8 carbons, and with only hydrogens. That's called a hydrocarbon, and that one's called octane. So gasoline is made up of hydrocarbons. Gasoline is not just one thing. It's got the octane hydrocarbons, and it has shorter ones with only 7 carbons or with 6 carbons. So that would be heptane, hexane. Pentane has only 5. These are the best ones though, octane, because they're longer. They have more energy in them. So when they burn, they produce more energy, so your car might be, you might get a little more power out of your car engine, a little more gas mileage with gasoline that has higher octane. Like 87 octane means that gasoline is 87% octane molecules. 89 means 89%. It costs more because it's better. But so this would be called the carbon backbone. And a lot of organic compounds have other stuff besides hydrogen. In fact, they will have some other small molecules attached to them, and those are called functional groups. Um, like a lot of, you'll see that a lot of organic compounds have phosphates on them. A phosphate is just a small molecule with phosphorus and oxygen. That's a functional group, a phosphate. Um, uh, Molecules like this, this is a straight molecule. These carbons are all in a straight line, but many molecules can curve up and form a ring shape. So this is the same thing, the same all the same atoms, but it's in a ring, so it's called cyclohexane. Just a different form of the same molecule. Glucose, sugar can do that. Okay, don't worry about these functional groups. When I took organic chemistry, I had to learn all the functional groups and how they react with each other. Organic chemistry is really hard. Organic chemistry is probably the most common reason that people don't get into medical school. I've known so many people went to college, majored in biology, planned to apply to medical school, but if you can't get better than C's in organic chemistry, you're not getting into medical school. One of my best friends wanted to be a doctor. Instead, he became a high school biology teacher, like it happens to a lot of people. He took the whole year of organic twice, still got C's. So
they got to take 1 and 2.
You have to? What'd you say?
One of the forms of they have to take 1 and 2, organic 1 and 2.
Yeah, that's organic's hard. Okay, carbohydrates. It's one of those 4 types of large organic compounds. So it's providing energy for cellular activities. So that's just because carbohydrate molecules like sugars are easy for your cells to break down and get the energy out of them. And the carbohydrates can be monosaccharides, disaccharides, or polysaccharides. Saccharide is Latin for sugar. Mono means one, so a monosaccharide, it just means one little sugar molecule. An example is glucose. That's what blood sugar is. And fructose, that's called fruit sugar. That's the sugar that normally occurs in a lot of fruits and also in corn. We all see high fructose corn syrup on labels of many kinds of processed foods. But those are small enough molecules that if you eat glucose or fructose, it doesn't have to be digested any further. It's small enough to be absorbed right into your blood, go to your cells, and your cells can use that. But these disaccharides, double sugar, that's two monosaccharides stuck together in a bigger molecule. And that example of that is sucrose and lactose. Sucrose is just what we call table sugar, like what you put in your coffee or on your cereal. That molecule is too big to absorb into your blood. It has to be broken in half. A sucrose molecule is one of each of these stuck together, a glucose and a fructose. So it has to be digested and broken in half, and then those monosaccharides can be absorbed into your blood. And what does the breaking apart of that is enzyme. There's an enzyme that in your intestine that breaks that in half. There's also an enzyme that breaks lactose. That's milk sugar, right? So one's in dairy products, except unless you are lactose intolerant. What does that mean? You cannot digest lactose. Your body doesn't make the enzyme to break that molecule in half. Um, so normally those sugars are broken apart in your small intestines and absorbed into your blood. But if you're lactose intolerant, that doesn't happen. The lactose just continues on all the way through your long small intestine until it gets into your large intestine. What do you have a lot of in your large intestine? Like living in there? Worms? Hope not. What do you normally have in your lot of bacteria, right? Um, they're called coliform bacteria because they live in the colon, or um, but they're supposed to be there. They're normal. In fact, you'd be in trouble if you didn't have them. Um, but they're not used to having sugar uh, coming through because normally the sugar gets all absorbed in the small intestine. But if you're lactose intolerant, you eat lactose, it goes into your large intestine. All those bacteria will have a feast on that. And uh, the the uh, um, the intestinal gas that is produced in all of us from time to time, that's produced by those bacteria. Um, and so when those bacteria feast on lactose, they produce a lot of gas and cause cramping and diarrhea and um, uh, unpleasant symptoms like that. Um, there are pills people can take if they're lactose intolerant. They can take those pills as they before they eat lactose, and it'll help digest it. What do you think's in those pills? It's just enzymes. Just enzymes. Notice these words end in OSE, glucose, fructose, sucrose, lactose. In biochemistry, OSE on the end of a word usually means it's a sugar. ASE on the end of a word means enzyme. It's an enzyme. So the enzyme that breaks down sucrose is called sucrase. The enzyme that breaks down lactose is called lactase, and so on. So those pills that you can buy if you're lactose intolerant, they're just lactase pills. Okay, so then polysaccharides, many monosaccharides in a long chain. Is also called complex carbohydrates. That's what starch is. In fact, starch is just a long chain of glucose molecules. So that's how a plant stores energy for later. Plants make glucose by photosynthesis. If they make more than they need right now, they put those glucose molecules together in long chains, and then they store that in somewhere in the plant, like a potato. It's just where that potato plant is storing energy for later in the form of starch. Animals like us do the same thing, but it's called glycogen. It's also long chains of glucose molecules. If you eat more glucose than you need right now, your liver will link them together in long chains of glycogen and store that in your liver. We also store it in our muscle tissue, store glycogen. And so then if you don't eat for a while, don't eat for several hours, your blood sugar is dropping, your liver will break down the glycogen into glucose and release that back into your blood to keep your blood sugar up and to provide energy for your cells of your body. If you eat so much extra calories that your liver is full of glycogen, can't hold any more, then your liver can convert glucose into fat, and then we store that on our bodies, and that's how we gain weight. Cellulose is also a polysaccharide. It's also long chains of glucose all three of these, they're the same thing, except that the kind of bonds in between the glucoses is different. In cellulose, we cannot digest this. We can digest this stuff, but we don't have an enzyme to break the bonds between the glucose and cellulose. That's plant fiber. So if you eat a lot of spinach or something that has a lot of plant fiber in it, we're supposed to eat that stuff even though we can't digest it. But it's good for our digestive system and stuff. But it just comes out in poop. Okay. This is just a summary here. This ATP, we'll talk about that later. Adenosine triphosphate, what that is. So monosaccharides, simple sugars, so there's glucose and fructose. Fairly small molecules. This deoxyribose and ribose, these are the sugars that are in DNA and in RNA. Okay, the sucrose and lactose and maltose, these are all double sugars. So we need enzymes to break these bonds when we digest this stuff. And that would be that to break that bond, that would be that hydrolysis decomposition that we saw a few years ago. There would have to be a water molecule present to be broken in half, and part of it would attach here, the other half would attach here when that bond is broken. Okay, starch, glycogen, cellulose, they're all chains of glucose. Each one of these is a little glucose molecule. Cellulose, it says the most abundant organic molecule on Earth because that's what trees are made out of, and plants are mostly cellulose. Every plant cell has a cell wall surrounding it made of cellulose. Okay, practice questions. Okay, we talked about carbohydrates, and they're only carbohydrates so far. So lipids, what they all have in common, they don't mix with water. So these are non-polar molecules. Fats and oils. Steroids are a type of lipid. Cholesterol is a steroid. Some hormones are steroid molecules. And phospholipids, this we'll see this again in the next chapter on cells. Phospholipid is a kind of fat-like material that forms a membrane covering every cell of every kind of organism, called a phospholipid. It's a lipid that has phosphate in it. But fats, we need to talk about fats. It's important. A fat molecule is called a triglyceride. And let's look at a picture of a triglyceride. So this is a fat molecule. Why it's called a triglyceride is it's described as having a head and three tails. The head part is called glycerol, and these three of these tails, so it's called a triglyceride. Three tails in the head. These tails are called fatty acids, each of these tails. And you can see it's just a long chain of carbon atoms with hydrogen attached. So what's the difference between a saturated fat and an unsaturated fat? The word saturated means something has as much as it can hold, right? So if a sponge is saturated, that means it can't hold any more water. But if a fat is saturated, it means it has as much hydrogen atoms as it can hold. And so what does that mean? Remember, each carbon can have 4 covalent bonds around it. Like this carbon, it's bonded to a carbon on the left, a carbon on the right, and then 2 hydrogens. So 1, 2, 3, 4 covalent bonds. This one on the end has only one carbon on the left, so it has three hydrogens. So all these carbons are loaded up with hydrogens, but look at this. These 2 lines here. That's called a double covalent bond. That means in between these 2 carbons, they're sharing 2 pairs of electrons instead of 1, and that counts as 2 bonds. And so these 2 carbons, because they have this double bond, and then they're bonded to another carbon, there's only 1 bond left. So it can have only 1 hydrogen attached instead of 2 like all the others. So this is what we call an unsaturated carbon, because it could have more hydrogen than it does if it didn't have that double bond. But because it has only 1 hydrogen, it's unsaturated. These are all saturated. It's only where there's a double bond between carbons that there are unsaturated hydrogens. If that's in only one place, like on this fat molecule, we would call this a monounsaturated fat. You've probably seen that on food labels. So that means only one place. If there's a whole bunch of double bonds, we call that a polyunsaturated fat. So if there were no double bonds anywhere on these fatty acids, that would be a saturated fat. So which kind are healthier, saturated or unsaturated? Anybody know? What did you say? Did you say it? You're not sure? It's unsaturated fats are healthier. Saturated fats are, what's an example of saturated fat? Butter. And animal fat, like lard, animal fats. Those are saturated fats. Unsaturated fats are mostly plant oils, like peanut oil, corn oil, olive oil. Yeah, and so saturated fats, like animal fats, are kind of solid at room temperature. They're kind of hard. But unsaturated fats, like plant oils, are liquid at room temperature. Which would you rather have flowing through your arteries? Something that's liquid or something that's solid? And why this would make it more liquid at room temperature, having a bunch of these. You notice these fatty acid tails are straight. This one has a bend in it at that double bond. And so if there was a bunch of bends like this, little zigzags in these fatty acid tails, they would not pack together as well as if they were all straight. If they're all straight like spaghetti, they pack together real tight. So that's what makes saturated fats kind of solid at room temperature. But because unsaturated fats have all these bends and kinks in the tails, they don't fit together very well, and so they're more liquid at room temperature. So saturated fats and also cholesterol they tend to build up in your arteries. So if this is an artery, this got this nice big open hollow space for the blood to flow through, saturated fats tend to stick to the inside of the artery, stick, and build up, and that's called plaque, right? But after years and years of eating cheeseburgers and pizza every day, that just build up more and more plaque, and this opening just gets smaller and smaller until there's just a trickle of blood that can get through there. And if that happens to one of the coronary arteries, which are the arteries in the heart itself, they deliver blood to the heart muscle itself. Blood can't get to the heart muscle itself, then part of the heart muscle dies. and that's what a heart attack is. And So a coronary bypass surgery is to replace that clogged artery. They'll take a vein from the person's leg or something and put that in there to replace the artery. They do something called balloon angioplasty. Now they'll put a long balloon into that artery and then inflate it to try to open up that hollow space bigger. If it happens to an artery going to your brain, it can cause a stroke, which is a stroke is an interruption to the blood flow to the brain. Either one can be fatal. If a chunk of this plaque breaks off and it's flowing through your blood vessels now, it gets into a narrower blood vessel and clogs it. Now it can completely block blood flow anywhere in your body, and that can happen in a lung and be fatal. Anyways, too much saturated fat is considered unhealthy. Hey, okay, we talked about this a little bit. Phospholipids are cell membranes are made out of it. Steroid molecules, they all look something like this. These four ring structures of mostly carbon and other stuff sticking out. So cholesterol and some hormones are steroids. Again, there's a fat, a head and three tails. This phospholipid that's in cell membranes, it's similar to a fat, but it has the same glycerol head, two fatty acid tails, but instead of a third fatty acid, it has this phosphate attached to it. So it's called a phospholipid. Okay, just a summary and a table about these kinds of lipids. Again, there's a phospholipid. We'll talk about that in the next chapter. Okay, practice questions. Okay, protein. So we talked about carbs and lipids. Now proteins, these are very important because there's so many different kinds of proteins and that do so many different things in our bodies. Some proteins are structural materials, like that means they make up some of the structures of our body, like this one called keratin. This one, anybody know where you have keratin?
Isn't that your hair?
Yeah, your hair and fingernails are made out of this stuff, keratin with a K. So we'd say that's a structural protein. You've heard of collagen. We have that in our skin. Our skin is made up mostly of cells side by side, but what holds that together makes it strong? We have these threads of collagen running through our skin to make it strong, as well as many of our other tissues are held together by collagen protein. So that would be structural. Energy sources, that just means we can eat proteins just like we can eat fats and sugars and break them down in our cells for energy. Some hormones are protein molecules. We saw a minute ago that some hormones are steroid molecules. Receptors on cell membranes, that refers to like receptors for the hormones. A hormone is a chemical it's a chemical messenger. It's like a molecule that has a message, and it's the way that one part of your body communicates with another. Our nervous system does that with nerve impulses, but a hormone is a molecule that's released in one part of your body and usually goes into your blood and circulates throughout your body, and it has some effect on cells somewhere else. So So we all know about the hormone adrenaline, right? It's called the fight or flight hormone. In an emergency situation, you have to either run for your life or fight for your life. That's what's meant by fight or flight. And so one of the things that adrenaline does, it makes your heart speed up, makes your heart beat faster. And that's because your heart muscle cells have receptors for adrenaline on them. So that is adrenaline flowing through your blood. If one of those adrenaline molecules comes into contact with one of those adrenaline receptors on your heart muscle cells, it'll cause that muscle cell to make your heart beat faster. But the cells of your stomach, for example, do not have receptors for adrenaline. Your stomach doesn't care about adrenaline. If you're running from a grizzly bear, it's not important that you're digesting food at that moment. So your adrenaline doesn't affect your stomach, but it does affect your heart. So they're called target cells, the cells that have receptors for that particular hormone. Antibodies are not alive. They're part of the immune system, right? But they're just special protein molecules of the immune system they're actually very smart molecules because they can search and recognize certain pathogens, certain disease-causing bacteria or viruses. So we have many, many different kinds of antibodies in our bodies. But each one is very specific. It's only looking for one kind of pathogen. Like you have antibodies for measles, and they spend their whole existence just looking for measles virus. They don't care about anything else. If they find a measles virus in your body, they'll grab onto it and try to disable it. We'll do a chapter. Well, you get that's next semester for you guys, anatomy 2, the immune system. And enzymes again. The enzymes are protein molecules that this word catalyze just means to cause something to happen or speed it up. In chemistry, it means to cause a chemical reaction to happen or speed it up. And these enzymes, do we have pictures of? Yeah. Talk about that in a minute. But so so those are some of the functions of protein molecules. But the structure of a protein molecule, it's like some of these other ones are made of long chains. These are very long molecules made of smaller molecules called amino acids in a long chain. And there's 20 different amino acids that make up all proteins. And so a protein molecule is just a long chain of these amino acids, and there's 20 different amino acids, and it's the order or the sequence of the amino acids along that chain that determine what kind of protein it is and what its function is. So these it's a long chain of amino acids, but I thought we'd have a picture. But the protein, this long chain of amino acids, and these can be hundreds or thousands of amino acids long, very long molecules. But then that molecule will fold up into a three-dimensional shape called a conformation. And that shape is very important to the function of the molecule. And I mentioned when we were talking about pH that enzymes are very sensitive to pH. If the pH of your body fluid starts to get too acidic or too basic, the enzymes will stop working. That's because what happens to the conformation is the protein molecule, the enzyme molecule will start to unfold and lose its shape. And when it loses its shape, it slows down or stops working. That's why, every protein has a favorite set of conditions, including its pH. It has a favorite pH. Some proteins, like the ones in your stomach, they like very acidic pH. If you put them in like pure water, they'll start to unfold. They don't like neutral pH. And then every protein has a favorite temperature that it likes. What do you think is a favorite temperature for the proteins in your body? What's your body temperature? About 98, right? So that's why if your body temperature starts to drop or rise too high, if your body temperature just goes down to 95, we call that hypothermia. And the lower it gets, the worse it is. But what do you think's happening to your enzymes? They're unfolding because it's too cold for them. Their favorite temperature is 98 degrees. It gets below that, the enzymes start to unfold. All those chemical reactions that are caused by those enzymes will slow down and stop. That's what happens to you when you're in hypothermia. Your body everything slows down. That's why we have to control fevers. We don't want a fever to get too high because it affects the enzymes. That's called denaturing, when a protein loses its shape because it's in the wrong temperature or the wrong pH or the wrong salinity or saltiness. One of the reasons should not drink seawater. Your body fluids will become too salty. That will affect your enzymes. Okay, so a little more about enzymes here. How do they work? I already saw this about the activation energy, but so here's a diagram showing how an enzyme works. This blob represents an enzyme, right? And so again, it has this characteristic three-dimensional shape of every enzyme, and that's the shape of this is like, it's like a keyhole. It has to be shaped like that because this this would be the enzyme sucrase that breaks down that double sugar sucrose. So there it is, a glucose and a fructose stuck together in that sucrose molecule. So if you eat sucrose, these enzymes in your intestines will grab that sucrose molecule. The sucrose molecule fits right in that keyhole in the sucrase enzyme, and then it'll break the bond. It says there's a water molecule because it's that hydrolysis decomposition. It has to be a water molecule to break in half for that to happen. But so then it breaks that bond, releases the two monosaccharides. Now this keyhole is called the active site. Now that's empty again. It can grab another sucrose molecule, break it in half, and release it. Grab another one. So the enzyme is not used up in the reaction. It's not one of the reactants. It can do this over and over, like hundreds of times per second. And without those enzymes, you could not digest sucrose, or could not do a lot of things. A lot of chemical reactions that go on in your body just would not happen without enzymes. And there's a different enzyme there's a different enzyme for lactose. It's got a different shaped keyhole that lactose molecules fit into. There are other kinds of enzymes will take two molecules together, two separate molecules, and join them together and form a bond and then release them. Okay, I guess we should take a break. Yeah, should we? Let's take a break. Turn itself off.
my sleep. My morning starts at 3:30 a.m.
What?
Yes, I drive to school, but then I go teach. And then I had a meeting today, and then come here. So when you see me like fighting, please just.
I hope every day isn't like that.
I know. That's why I got some coffee today. I was like,
A lot of coffee for a day like that.
them be home tonight at 12, 1 o'clock, saying I got to wake up at 3:30.
You get up that early to ride the bus every
Yeah.
day?
Cause our bus is like an EC bus, so I'm from Anson County. We only have like one school that does like the EC, the special, real special. So yeah, so we go all across the county. I get up at 3:30. I leave my house like at 4:30, 4:55. Get my coffee on lot 5, 4:50, 4:55. Pulling out at 5.
So you start picking up those kids that early? They're out there
Our
ready?
first pickup is at 5:30.
Wow. Hmm. Hmm.
So like, since I broke my leg, I broke my tibia.
Hmm.
Yeah, my tibia. I broke my tibia and I tore my meniscus.
When was that?
2023. So that's why sometimes you'll see me dim.
Hmm. Your meniscus doesn't heal right?
I got a second opinion, and he's gonna do a MRI and see, cause he said he don't think like it when he extend my leg out. He said something don't seem right. So I'm waiting to go to the doctor, see another doctor to have this second opinion. Cause when you get hurt like that, you just get thrown to the ER. And I mean, I followed up and everything, but the doctor, the surgeon that did my leg, I would not have chosen him.
Yeah. He's just the one who was on call with the ER
Yeah.
that time.
They sent me to CMC main trauma.
Hmm.
Mm.
Sometimes for a badly torn meniscus, they just remove it. And that's what they used to do. Um,
He said it was really bad, like in the middle, the medial. That's where it's torn, and he said within like maybe 5 years I need a knee replacement.
Yeah, they used to do that when football players would get their tear a meniscus, they hit the leg sideways, or tear a meniscus. Doctors just take it out. Because
Take it out.
it
That's the cushion.
it would just take too long for it to heal, or because cartilage doesn't heal very well or very fast, and that guy player, your bones have their own articular cartilage on them, but without that meniscus, like you said, that guy can play a few more years, but if he's a professional football player, but 10, 15 years, he's going to need a replacement. He's going to have arthritis so bad. I think now they don't as readily remove them. They try to save it.
Mm-hmm. That's what he said. They stay for. They try to
Yeah.
preserve it. So I said either I want to be an orthopedic nurse or I want to be like a cardiologist. I couldn't take anything that is great.
Yeah. I had a friend who became a veterinarian, went all the way through vet school, became a veterinarian, and she couldn't handle it emotionally because whenever a dog would come in and die, or people bring their dogs in to be put to sleep, she would just fall apart, and she couldn't take it, and so she never really practiced.
So she made it.
Yeah, but
Emotionally,
she couldn't handle it, so she never worked as a vet. All
Because
that.
death is natural. You know, it's
Yeah.
natural, but still, it's hard to accept.
Yeah, I can understand why.
Yeah, I can too.
Yeah. Yeah, she ended up teaching teaching college. That's how I knew her. At
You met her.
when I was at Coastal Carolina University, she was working there.
So next week we have our exam. We'll just go back, we'll break, or we're gonna leave after our exam.
Oh, I'd like to say that we'll just leave, but we'll have to come back and do some other stuff. Can't really afford to waste that time.
I know.
And I'm not supposed to do that.
I was just asking. Online. You got your laptop too? Is there a copier in this building?
No,
Okay.
not that we have access to.
Okay. Yes, I know. I'm dreading chapter 2. Oh, okay, it's gonna take me a minute to understand it. By the time I understand it, there's gonna be test time. Because I don't like chemistry. And probably have to take chemistry to get your BSc, and I would imagine. Yeah. By then, maybe I have a little bit more subjects. Yeah, because I had a meeting, you know, my teacher had a meeting. You did? And that's what
Oh, her thing, she had COVID.
Oh, Lord. Yeah.
Did you take roll today?
No, I didn't. I gotta do that, thanks.
Right, because I need all my credit. I'm gonna be in here, I need my credit.
Yeah, because I found out, I was like, I want to get you here.
She texted me, but yeah, I got here about 5:45.
Next Monday.
We got the test, we definitely need to be here.
Mhm.
Monday.
I think that's teacher work day for us, so, I keep here.
Okay.
And then he put that word right back in the head. And I need to get a book, but I'm scared the book's gonna change. And I was gonna sign up for two, but
I have a
some,
book.
yeah. Can I see it? Oh, wait a minute, let me show you the tip first. I think it's bad one too. I'm saying, I'm nodding now. I'm looking in there. I'm about to fall asleep over here. Make sure you don't eat that. You should. I'm gonna let you type it in. It'll be torture. get you.
Right. Perfect. Thank you so much.
I'm gonna do this too.
Okay, yeah.
Both of them.
Yes, thank you. It's right there. It's on there.
No, I'm finna do the other one too.
I'll just get the attachment and right here.
I'm sitting here rushing.
You all right? Yeah, okay, I'll take you. All right, thanks.
You're welcome.
Okay, let me ask you a question.
They,
Yeah. Okay. I teach. Okay, we all.
I'm gonna use this one.
Let me see if I can have this.
It's cold.
Yeah. I'm like, dang. I thought it was just me.
No, honey, it's all the same.
Oh. Black markers. Ah. I think you close it. Yeah, I think the ball back.
Sorry.
Ah.
If I find my old anatomy book, I probably can use it, can I? It would probably be the same. I wouldn't think.
When
much.
was it?
It was a white book.
Uh,
I know I didn't sell it.
it's not the same, but they all have the same stuff in them.
Okay.
Okay, so we talked about carbohydrates, lipids, proteins. The last type of large organic molecule that's important in our bodies is called nucleic acids. And that's what DNA is, DNA and RNA. And these are also very long molecules. In fact, one of our lab activities I got to do tonight is really cool. We're going to extract DNA out of strawberries. Have any of you done that before? It's cool. Actually get the DNA out of strawberries, separate it, and the DNA molecules are what chromosomes are made out of. Every chromosome in your, or almost every kind of cell in your body has the same 23 pairs of chromosomes. And those chromosomes are very long. Some of them are like an inch or two long. These huge, long molecules, but they're all coiled up inside of every cell. And so these are long chains of smaller molecules called nucleotides. We'll look at what a nucleotide looks like in a minute. But DNA is short for deoxyribonucleic acid. RNA is short for ribonucleic acid. When we were looking at those monosaccharides, there was the picture, the diagram, there was glucose and fructose, and then on the right there was deoxyribose and ribose. Those are the sugars that are in these nucleic acids. That's why this is called deoxyribonucleic acid, because it has a sugar called deoxyribose in it. This has a sugar called ribose in it. So this makes up chromosomes. Chromosomes are how our cells store information. So each chromosome is a very long DNA molecule, and the DNA stays in the nucleus of a cell. The nucleus of a cell is a big it's a membrane that surrounds the chromosomes, and the chromosomes stay in there. But each chromosome, if this was a chromosome, then the chromosome was made up of a bunch of short sections called genes. A gene is just a section of a chromosome that has some information for the cell to do something, and usually what that information is the instructions for the cell to make a particular type of protein molecule. So cells are always making proteins, especially enzymes, because there's always chemical reactions going on in cells that require enzymes. So if a cell needs to have a chemical reaction happen, it'll make an enzyme that'll cause that chemical reaction to happen. And then after it's done, doesn't need that enzyme anymore right now. It'll just take that protein molecule apart into those separate amino acids, and then they'll be reused again to make a different protein. But the proteins are the chromosomes stay in the nucleus, but the proteins are made elsewhere in the cell, out of the nucleus. So how does the information get from the nucleus, where the chromosomes are, to out in the cell where the proteins are made? It's with this stuff, RNA. So if the cell is going to make a protein using the information in this gene here, this one, this chromosome has to stay in the nucleus, but the cell will make a copy of that gene that copy will be RNA. And so think of the chromosomes as like books in the library. There are some books in the library that you're not allowed to check them out. I think they're called reference books, I think. They have to stay in there. You can't take them with you. But you can make copies of some of the pages of those reference books and take those copies with you. That's what this is. It's making a copy of that gene, and then that copy in the form of RNA, that can leave the nucleus and go out into the cell and be used for making protein molecules. So what do these nucleic acids look like? They are long chains of smaller molecules called nucleotides. In fact, these nucleic acids, another name for a nucleic acid is a polynucleotide. So it's many nucleotides in a long chain. and there's only four nucleotides, four different nucleotides that make up um, a DNA molecule, and you don't need to learn these words. Adenine, thymine, cytosine, guanine. We just go by the letters A, T, C, and G. So there's kind of like the letters of the alphabet of the language of genes, the genetic code. And so along a DNA molecule, it's just a bunch of letters in a certain order, and that's how the information is stored. Um, so, well, for example, along this, there might be just A, T, C, C, G, A, T, 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 G, A, C, C, and so on. That's the information. Um, the cell is able to read that and translate that into which amino acids to put together in what order to make the protein molecule. Um, um, so here's two different types of diagrams of DNA. They're both trying to say the same thing, but they look different. Two strands of nucleotides side by side. So this is a nucleotide, this is a nucleotide. So every nucleotide has three parts. There's that sugar, deoxyribose sugar, and then there's a phosphate. Remember, there's that phosphate, that's a functional group that's in a lot of organic compounds. And then this one, though, is that letter, where the letter is. That's called a nitrogenous base, this part. But so we would just call that the whole nucleotide T. And this one is the G nucleotide, and A, and so on. But there's two strands of nucleotides side by side with the bases toward each other, the T and A toward each other, and those are hydrogen bonds, just like those weak attractions between water molecules. So these must be polar molecules to attract each other like that. And those hydrogen bonds are what holds the two strands of DNA together side by side like that. Where there's a T on one side, there will always be an A next to it. G will always have C next to it, so there should never be a G next to an A or a T next to a G or C. So we say that T and A are complementary nucleotides, and G and C are complementary nucleotides. They're always together in pairs like that. So this is another diagram of DNA showing the same thing. So nucleotide, two nucleotides. This might be G and C, T and A, or something, and the whole molecule is twisted like a ladder. It's called a double helix shape, that twisted ladder shape. RNA is just one strand of nucleotides, and each nucleotide again has a phosphate, a sugar, and a base, and these would be also, there's only four of them, but instead of A, G, T, and C, it's A, U. It's A, U, G, and C. So in RNA, there's no T, there's a U instead. So this would be A, U, G, or C, and so on. I think we'll talk a little more about DNA in chapter 3. But, so that's comparing the two, DNA and RNA. So A, G, T, and C in DNA, but A, D, U, and C. So there's U instead of T in RNA. DNA is 2 strands of nucleotides. RNA is just 1 strand. DNA is twisted. Into a helix. RNA is not twisted. Okay, and finally, just one more little molecule need to mention. Now we'll be done with this chapter, but it's very important. This little molecule, ATP, one of the most important molecules in all of biology, because every cell of every organism we know of, from humans all the way down to bacteria, they all use this little molecule like rechargeable batteries for storing energy. So when a cell breaks down a sugar molecule or something to get the energy out of it for the cell to use for whatever it does, the cell won't necessarily use all that energy right away. It'll store that energy in a bunch of these little ATP molecules, like little rechargeable batteries. Then whenever the cell needs energy for something, it'll use the energy in the ATP molecules. And ATP stands for adenosine triphosphate. So there's that phosphate again, like that keeps showing up in organic molecules. So here's what it looks like. This part of the molecule is called the adenosine part. But then here, that's a phosphate, that's a phosphate, that's a phosphate. These squiggly lines represent bonds holding these phosphates together. And so it's called ATP because it has 3 phosphates, adenosine triphosphate. If it has only 2 phosphates, it's called adenosine diphosphate. Adenosine diphosphate is a dead battery. Adenosine triphosphate is a charged battery. So how this molecule stores energy is in that little bond right there. The 3rd phosphate bond on ATP, that's like the most important bond in all of biology, because that's where all of the energy is that keeps every cell in the world alive. So whenever a cell needs energy, it can take that 3rd phosphate off, release that little bit of energy, and use it for anything, for making proteins, making my muscle cells contract. Anything the cell does, it uses that energy. So these are constantly being the energy being used, and then they're being recharged, and then they'll use the energy and be recharged. So you're inhaling oxygen right now for those reactions in your cells to break down sugar and other nutrient molecules to get the energy out of them. Then that energy is used to recharge ATP, and the ATP is used by the cells to stay alive. If you stop breathing, your cells stop recharging ATP. All of your cells have enough ATP to keep them alive for just a few minutes. If they can't be recharging those ATPs, it's like all those batteries go dead. And when that happens, the cell runs out of energy, it dies. And when your cells die, you die. Your whole body doesn't die at once when you die. Some cells live longer than others, but if your brain dies, brain cells die, and they're usually some of the first ones to run out of ATP and die. Cells in the rest of your body might be alive, but you're dead if you're brain dead. So I'll be mentioning ATP many times through Anatomy 1 and 2 because it's so important. Okay. Well, I rather than start chapter 3, I don't feel like it. I'm tired of talking, and rather do some let you guys. I know everybody's tired, but we can get up and do some fun lab stuff. And we can play with this guy if we want. Let's do that. This is I just played with this a little bit. I spent a total of maybe an hour on this. But this thing is really cool. the tilts, normally the bed lays down, so you can use it that way, or you can tip it up like it is here. And you can't really roll it very easily to face you guys. But there's different cadavers, different there's I don't know, several dead people stored in here, and this one's name is Carl, and it even has their medical history in here, and you can see what he died from, and some of them I was looking at one a lady cadaver in there, and she died of liver or stomach and intestine cancer. And you could look at her stomach and intestine and see the cancer. It's really cool. Not for her, but she wasn't. But some of the things you can do. Let's see. Where was that? I learned stuff and then I forget it. So anyway, you can take sections anywhere in any direction. You can find the function I'm looking for. There's one where you can choose organ systems. Like you could make everything except the skeletal system disappear, or everything except the reproductive system disappear. But I can't. How did I do that?
That's not it in that little box.
This.
Uh-huh.
Well, maybe. Well, this might be one way to do it. Let's. Okay. So yeah, that's shows his digestive system. So the esophagus that he swallows through, then the stomach, and this is all small intestine and large intestines and rectum. Where was that actually? I don't keep finding.
You right there.
Is that it?
Uh-huh.
There was you just touch them and it'll highlight it and label what it is for you. So that says transverse colon, descending colon, and then you make that go away. So things like that I don't know yet. There's a lot to learn on here.
There's another section that popped open right there.
Oh, here. Yeah, this is it. This is what I wanted. Yeah, the eyeball. So these are a checklist for the organ system. So if I uncheck all of them except this skeletal, then it's only the skeleton. Now, where's his legs and arms?
It wouldn't be up. You see up top, it's like a beside that star. What's that?
Add to subject. I don't know.
No, beside the star. To up top, it looked like something in the hat.
In here?
No, to the left. What's that? No, go back. Right there. What's that?
That's a hand.
No, right there. What's that?
Actually, I don't know.
That's all.
Okay. You can come up and play with it if you want. You got anything you want to play with it? You're welcome to. I think we ought to spend some time with it and learn about it. Well, so, yeah, there's the cadavers. Yeah, let's look at that. So there they are, the different cadavers. There's Victor and Vicky and Hans and Carl and Carla.
Which one had the stomach cancer?
It was one of the ladies. It was Vicky. And so down here, you can check you know, what body parts you want to look at. So where would this stone intestines? What's the third one? What's the rest?
I thought it was like up on my body.
Yeah, you know, let's find where the natural history is. See what's wrong with them and then we can look at those. So you can see what is it. There's the brain and spinal cord. So I guess it's have to reinstall, re-upload first, and then
Is that what a cancer is?
I think that is, yeah. See if we look at her only her digestive system. Let's see if she's the one. Remove this whole thing. Got those fat away. Yeah, greater omentum, lesser omentum, that's a layer of fat in the crop. Make that go away. Okay, so yeah, I think that's her diseased intestines there. Did you see that?
Mm-hmm.
Yeah, it looks different from the rest. I think that's what killed her, stomach cancer, or intestinal cancer. But it can rotate.
How old was she?
Well, I don't know, where can we find their histories? Patient history. Somewhere it tells all about each cadaver. Um, somewhere. Yeah, it also has animals, cats and rats and dogs. Mhm.
What's that?
Resolution. Just, if I'm going to take that for something. Yeah, I'd like to know more about these people.
So only one female,
Part of us,
Oh. You call
there's part of them.
Oh, Carla. Oh, well, they friends, I wonder.
Anyway, I got this, well, I want to do this chemistry, uh, chemistry stuff. So there's a couple of things, why don't you guys head over here, okay? Everybody step over here. Yeah. So there's two setups here. This one is for extracting the DNA from strawberries. There's strawberries in here. I got at Walmart. I'm going to eat what we don't use. So there's the instructions here tell us what to do, but we get Ziplocs. We got Ziplocs, so you just put a strawberry in a Ziploc bag, and you just squish it up, mash it up with your hand, and you just turn it into mush, and then you mix it with. Do you leave it in the bag or what's it get? Yeah, I guess you just pour some of this stuff into the bag, a certain amount, and then you just mix that in the bag. And then so we're breaking the strawberries up, and this stuff is it's mostly just dish soap and salt. Um, and so we want to get the DNA out of the cells. What are the cell membranes that surround the cell? What are they made out of? Phospholipid. Lipid is like fat. What does detergent do to fat? It dissolves it, right? So that's what this is for. It's just to dissolves the cell membranes and the nucleus membranes to let the DNA out. And so then we take a funnel, take some of this cheesecloth. This is a filter. We filter this mix, put that in there, and then you dump your stuff in there and filter it into a test tube. And so there's going to be lots of molecules from those cells that end up in the test tube. But what's different about DNA is DNA is very, very long molecules, like an inch or two long, that are going to be in here. And then, so when you're done straining that, then you take, using one of these droppers, I've got some ethyl alcohol in the refrigerator in the other room. I need to go get it. It's supposed to be cold to make this work best. So, so you have maybe an inch of this slop in the bottom of here. Then you carefully dribble that cold ethanol in there, just down the side. You don't want it to mix in. You just want it to be a separate layer on top. Um, and after you do that, you get maybe an inch of ethanol in there, and the DNA will come up out of the bottom of the mixer and the bottom. It'll come up into the ethanol. And so, and you'll see it. It will look kind of cloudy. Look like little long strands of, it looks like snot in there, whitish snot. And then you take one of these glass starting rods, and you just carefully put that in there, down to where the DNA is, and you just slowly spin it, and the DNA should wind up on that, onto the glass rod, and you pull it out, and you'll be hanging off of it. Long DNA molecule that look like snot hanging off of it, but that would be pure DNA. Really cool. So anyway, and then this one, it's a more complicated little bit. Well, not really, I guess. But this is, it says, this is biological molecule lab, and it's we don't have much time. This is for if you have a solution and you want to know if there's any protein in that solution, you can add this stuff from a Bioret reagent to it, a Bioret solution, and it'll change color if there's protein in it. And it tells us on here what colors it changes. It changes from blue to violet or purple if there is protein. If there's no protein, it won't change color. And if you wanted to know if there's starch, you have a solution, you want to know if there's starch in there, you put this stuff, this iodine like people put on cuts. And if there's starch in the solution, then it changes from the normal brown-orange color to dark or very dark purple. And if you want to test for monosaccharides like glucose or fructose, you use this one called Benedict reagent. And if there's monosaccharides in the solution, it'll change from blue to green, yellow, orange, or reddish brown. It doesn't change color for sucrose, disaccharide, only for monosaccharide. And so these are used a lot. And you ever watch that Forensic Files? You know what that is? I love that show. Where they use forensics to solve crimes. There was one episode where they used these exact tests. They were on the stomach contents of a murder victim to find out when his last meal was, based on what he had in his stomach. And from that, they were able to determine that some suspect was lying about when he last saw this person suffering. So that was used as evidence. But like if he had eaten somebody, if you ate a hamburger for dinner, there's a lot of protein in it. But if your last meal was breakfast and you ate cereal, that's mostly starch and sugar, right? No protein, hardly any protein in that. So that's the kind of thing they can use this for. But anyway, so I got a bunch of things here to test. Got some potatoes, just mashed potatoes mixed with water. Got milk. Got this stuff called albumin. It's really egg whites. Vegetable oil, gelatin. I'm using that gelatin and water like Jello. Glucose and corn syrup and this little diet. So this lab activity is just to use these to test some of these to see what's in them. So, Nita, I get it, the fastest way is just divide it up into groups, divide it into groups, and one group do the test for sugars, one group do the test for protein, one group do the test for starch, and you just need a test tube holder with 4 test tubes, and it tells you what to put in, like for the one test for sugar, you're going to test the potatoes, the glucose, and the corn syrup. And the fourth one, you just put distilled water in there. And so you put them in there. Same thing for the iodine, you're going to test for potatoes, glucose, corn syrup. The protein one, you're going to test egg whites, milk, and gelatin. But it's simple, you just put it in, put the 3 drops in there, and see if it turns color, and just write the results in the little chart. But the one for sugar, for the beverage solution, you have to cook this one. You put the stuff in the test tube, and then you just have to put it in boiling water for 3 minutes. So I got a hot plate, and can you plug that in? And I'll put this in a microwave to heat it up faster. You just put the test tubes right in there, leave them in 3 minutes, take them out, and you're done. There's one more test though, for the grease spot test. And that's this one's highly technical. It's a piece of brown paper bag. The test for lipids. So how would you test if you have a solution and you want to know if there's oil or fat in that solution? You smear some on paper. What's that do? What does oil do to paper? It makes a grease spot, right? You hold it up to light, the light comes through it. If you do that with water, it's not the same. It might get it wet, and it might look a little like a grease spot, but it will evaporate and disappear. A grease spot is smaller. So I got the other hot plate there. We could turn that on low so that when we put the water, we'll just put oil on one side, water on the other, and then put it on the hot plate to make the water evaporate faster. So that's all there is to that. Then you just write on there oil on one side, water on the other, water, put it on the hot plate, and see if it leaves a big spot. So there's still a few attendants, but 1, 2, 3, 4.
When we get there, I need credit.
So there's 12. Let's do 4. Let's do 4 groups of 3. So one group can do the DNA. One group can do the sugar, the one you have to cook, and then one group can do, I don't know, do the protein, and another group do the starch, and then so many of you have, if you get done, do this, just do this. Whoever gets done first, do this one. Put this all in a minute. Okay, so should have what you need here. I gotta go get the cold ethanol for that. I'll get that water boiling. But, so, I don't know, you guys wanna form your own groups, or should I? Anybody wanna do DNA? You
We're doing it, we're doing it, we
need one more.
Ashley, come on.
She's over there.
You got to be in the group.
Okay. You three got more groups?
Where I'm at.
So, who wants to do sugar?
Not a sugar.
Virus is for protein. You guys want to do protein? And then for starch. So you just need a test tube rack, four test tubes, the droppers and
You
everything.
want over here listening, so I don't know you going over there. You go there trying to see naked people. You said naked people. Right.
And two.
What do you think?
A small piece of
I take it potato and potato.
We're in North Carolina.
Fair.
She just said a small piece. So this
This one is not a. Okay, boom. Make up, boom.
No, after this.
Okay. Hey, you don't get the stuff, the alcohol?
Huh?
The alcohol?
Yeah, I'll go do that.
All right, stop.
It just said a small piece.
Okay. Hold on. Okay, we got that. We'll start this slurry. Yeah, one more person down in front of you. Is that not station too? Oh.
We don't have to see. And a dropper full of food.
Hey, what are the groups? You guys?
Yeah, I thought it said.
Group could be So what, so I guess you'll be doing the protein, that's for protein, so you need one of these
Just a whole dropper full.
and a handout. If you would.
Dropper full.
So look for the bio, that's for protein. That tells you about the color.
Okay, 2, 3,
So,
and
in
a
your
dropper
four test
full.
tubes, you want one of them just,
I know, that's what I'm saying, I say, you know.
one. So, it's just a drop of full of these, maybe 2 drops.
A full dropper.
And then one, and then the last one is just water.
And 2 drops.
What's her name? Who? The other lady that found. Crystal, okay. I was wondering, you know.
Okay, and 2.
You wanna take roll, Mr. Beck?
And
Yeah.
a dropper full of.
We here.
So after everyone's done, we'll,
Do we have to do all this down in there?
no,
Okay, I'm about to wait.
just so that the dust gets away.
And then I place all 4 tubes on a boiling boiler for 3 minutes. And then after 3 minutes, I'm sure you already know what it is.
Right.
Yeah, there's solid.
Is that what we're using now?
Yeah, it has to have the same password and then you put that inside.
Yeah, because all this, you don't need all that solid stuff.
Oh,
How do we do a water bath?
Yeah, 'cause the water's not here.
Oh, it's in the microwave.
Oh,
Yeah, it's online. Okay.
yeah.
It's already filled.
Oh, yeah, I mean, I
He told me, he'll, if I haven't heard from him in a week to email him back to her.
Who you talking to?
Mr. Brown.
Yeah, I signed up for it already. I signed up for this.
You are?
My hands are cold. It's cold in here. Nice little warmer.
What's supposed to be like rotation?
Which one, the nurses? Yeah.
Well, I was asking Sean, get a program.
Again?
Okay. And she said I was on the right track. I just need to get there.
Did you say? Okay.
Okay.
How was it?
It was nice.
I haven't eaten. It was not bad.
I have to take one to bed.
I don't know if y'all, is there like that you do a person test?
Just find 3 minutes.
And then one person.
You have to attack it.
Look it up first.
No, no,
I don't know.
Oh,
Yeah, I already did that now, because I went to, physical tech for family. I already have a degree in, I did that.
So you did that over there?
Before I
I
came.
got a degree, so I got my 3 degrees.
Look. And
Yeah, so just write what's in each group.
You have to go like all
Yeah.
the other night.
I wanted to,
Write in the colors. My color ones,
Because you have to get up whenever the body comes.
I did my money in it.
I can't tell the difference between brown and
You work somewhere else.
I was looking at it on TikTok.
Sometimes.
Like they have to get up.
So I did, I was mobile
And on the shade. But-
and.
So if that's brown, that means it did not temperature color. So that
You know, the area.
did any of them change
Like,
color?
there, you know, this side, you go across the way, okay, and you go get a more.
I'm glad they did that.
Did those change color? Did you like them?
Yeah, I did. Yeah, I did. I got different ones.
But, you know,
Yeah.
oh, yeah, that's it right there.
It's a whole lot snot. I be stirred.
Would you expect everybody to
You can see they fix it.
No,
Doesn't change color?
No, this is funny.
All right, fire.
That's it, right there.
What's not in
Right there.
That's,
I don't see it.
Wait a minute, stop for a second. This is it, there. This is it, right there.
I got that.
Y'all
I really do. I've been out of work. I never heard it work.
You gotta sit right there.
Yeah, I had a tear. I do too. And this side. And I'm on work phone right now.
You'll get fixed.
No.
You don't like
that? It's a black, like, sir, and I'm just gonna stay there. I'm just trying to come to job.
They gonna pay you, but you can fix it.
Why?
I'm scared.
It's
Never, I ain't never.
It's never gonna, like, the
Be the
spot,
same.
right, like, why fix it?
It ain't never been said.
Yeah, so what I'm thinking is, right here, I just like all of them. I just think so.
So when you was out.
But it did this same thing last Friday.
Let me show you what I got her. You like it? You like it?
I think I see some.
A whole bunch of bottles.
Yeah, that's it.
Yeah.
It's right here. Yeah.
Listen.
I'm trying to be funny.
Yep.
That's
I can't see 1, 2, 4, 5, but I get what y'all saying. About 5 minutes. She had to just put it right there. Mr. Beck, did you see it? Yeah, we already pulled that.
So that's it. That's
Yep.
DNA.
It's right over there.
You do see it, put the number 2 here, and what color your picture was, so you got.
You want us to clean this up?
Let everybody, anybody want to see the DNA that they got?
Everybody for coursework and
Doesn't look like much. So that's DNA mounted on it.
Oh, it was like, okay. Ain't it on the report?
Yes.
And you have to take one each.
Yeah.
We took on one of them.
Dang, so that's gonna be a long time. Yeah. Oh, it is.
It's on more because it's so.
It's not going to but they take them, well, she said she took like 100. You take 100, sure, yeah, 100, that's 150 when I asked her about that and. But you got your degree, so that's gonna push you up. Points. Yeah, so your points gonna push you up.
That's gonna, I have a bachelor's degree and all the associate.
Yeah, all that's gonna push you up.
But at your point,
No, but we're gonna share
We're
the
gonna get
results.
it,
when
yeah.
they're all done. We'll just see what everybody got.
I need to get in that program.
That's what I want to do. I want to then I want to do RN, but then she said,
Right. If that don't work, then I'm just gonna go to USCP and do my BSc, because this class is a transfer. Yes. The USCP, yes. I am, I'm taking this one, two, both of them.
I'm on the degree. That's gonna get a lot of that degree.
Yeah. I don't wanna go back, but I'm on leave right now because of my, and I just, like, you know, I just don't wanna go. I don't think, yeah. I gotta work something.
You gotta work. I like to. Yeah, that's pretty good pay. They take a couple years.
Right.
To get paid.
Yeah, yeah, I
You
got
gotta get paid, don't worry about it.
Right.
Take time to get.
Yeah.
I'm working.
Oh, you're going.
No.
Maybe. I ain't even gonna try this.
It's almost 8:50 now, come on here.
Maybe next year.
Look, I don't need to this.
Next year when I'm.
I need to get in there. And like when I mentioned, she's like, if you're gonna try for all this, you need to go ahead and take this. I was like, okay.
Yeah, on the summer, I went ahead
I
and
said,
put
sign me
it
up.
I signed up too for it. I was like, Lord, I don't know about no online. When I was still in school, it was all. I mean, well, that's my bachelor's on the online. We need to. My bachelor's community is all online.
If there is.
And I'm not used to Aaron. You know, I know, and I know that.
And then you know it's gonna be fast track in the food.
I was like, ooh.
Okay, real quick, you, we did proteins, so what did you get for results?
The, yeah, some of the egg whites changed.
So the milk, gelatin, and egg whites, all three of them have protein. Gelatin is pure protein. Egg whites are protein, so that, we expect that to all be positive. And for sugar,
It was
all positive, so we tested what, glucose, corn syrup, and
Water and
glucose, corn syrup, and there was another
water and potato.
potato. Okay, so they all they all had monosaccharides, simple sugars. And starch, that's you guys, right? So what did you get for, did you go test potatoes?
Yeah, we did
Was
potatoes.
that positive? Or-
We had positive for that. It's kind of like a brown
Yeah.
purple.
And then
color.
cornstarch,
Yeah, it was like 1-3 positive.
cornstarch should be positive for starch, right? And what was the other one?
We had glucose, it was like a yellow, and that was positive. And then water, yellow, positive.
So you got positive for starch and glucose. And that's, I'm surprised, because they're, but, you should not be starch and glucose, but I believe that sometimes we can get the results we expect. So, the grease spot in water, it's on the hot plate, I'm sure the grease spot made a grease spot, or the oil made a grease spot, the water didn't. And then the DNA, you guys got some DNA on those glass rods, right?
Yeah.
So that worked okay? Okay, I still didn't do this kind of stuff. Graceland, Twinkle, didn't show up. Mary, LaTracy, Everett, no. Kelly, Hannah,
Here.
Kahela,
Here.
Tiana,
Here.
Sherry, I didn't see Sherry, nothing. Shania, Ashley, Sharonda,
Here.
Shaquala,
Here.
Kaylin, Kaylin, that's it, and Tisha.
Yeah.
Okay, let's get out of here. So exam next week, first two chapters.
All right, have a good night.
See ya.
I know what's going on. And I know that we don't want to have 50 questions. Is there stuff that's on here that's actually really not going to be on
When did you print this?
there? At home?
Today or yesterday or something?
No, yesterday.
Okay, but I forgot this was on it, and today I looked at it and I deleted some stuff that.
Oh, okay,
that
okay.
you-
Well, I just go back and
like,
look at it.
What did I, see if I can remember what I deleted. There's some things that we didn't cover.
Mhm.
Let's see.
Because I do better when I'm hands-on.
Yeah.
Reading is like, okay, what the heck are you talking about? And then like when I go home, I'm gonna have to go all back over all of this
Yeah.
and try to do stuff that's more hands-on.
Each have our own learning styles and different kinds of intelligences. Don't worry about this, that's not gonna. Um, think I lost out a few things on here.
No, I just did the ones, I was going over the ones I knew for a fact. Yeah.
Um, I don't think this stuff was on the PowerPoint. It's a lateral and frontal, so I don't think that'd be the same. Yeah, don't worry about any of this stuff. And I would say the sample questions are more important than this.
And that's what we took online, or is it?
No, not the quizzes that you took for a grade. The sample questions that are on Moodle right below the PowerPoint.
Okay.
Print them out and test your knowledge with those.
Yeah, because I print like 4 different packets. I do them, and then I check them to see which ones I got right. And the ones I get wrong, I go back and make sure I study those more.
Yeah, look that stuff up, and
Yeah.
that's I think a good way to study.
Because my kids think I'm crazy, because I be in the house acting as if I'm a teacher, teaching what I learned to
Well,
make sure I got it right.
Teaching is the best way to learn stuff.
Yeah.
In fact, I never knew biology very well until I started teaching it. Now I really know.
Yes. So yeah, thank you so much.
Okay.
You have a nice night.
Come on. Come on.